As already introduced, I want to focus on a few aspects that we evaluate as being crucial for strain engineering for biopharmaceutical production. Can give it a click. So within our global company, just to get you a view where we are located, my lab is located in Frankfurt in Germany, and we are part of our global CMC development unit called microbial platform. So as you can already deduce from that, we are dealing with microbial processes. And for those of you who are not familiar with the expression CMC, so that stands for chemistry, manufacturing, and controls. And that's related to a section within a regulatory dossier for new drug application, which basically explains how the drug look like, so its chemistry, how it is manufactured, how the process looks like, and also what analytics is done to ensure quality control. And so our job is basically we are getting innovative assets, new molecules, that we are converting into a commercially viable product. And the core of that is process development, covering basically strain engineering, fermentation, USP development, and also purification, DSP development. So here's a little overview on the assets that we as Sanofi are working on. So I've highlighted the ones that are produced in microbial expression systems, or for that at least some precursors are produced in microbial expression systems, but in the talk now I will focus on our nanobody molecules, as you heard already yesterday. So this is a love relationship, Pichia and nanobody, and this is also what we are working on together with the colleagues from Sanofi Ghent in Frankfurt. So bringing that into the clinics and hopefully also into the market. So for process development within CMC, we have some goals linked with that we already want to tackle at the strain engineering stage, as we think the basis of a great process is a great production strain. If you're not gonna solve some of the issues already at the strain level, it's gonna be challenging to deal with them afterwards during USP or DSP. So some of the typical goals that are linked with strain engineering is high productivity, a driver for COGS to a certain limit at least. We want to reach suitable product quality. We want to have documented provenance and clonality documentation for the strains that we are using for commercial production. We want to ensure genetic stability of the production strains, and there are also some aspects associated with strains that support process aspects, as for example, facile and robust processability. And also on the strain level, you can already influence that you have eco-friendly or operator and safe facility-safe processes. I've given an example already at the last Pichia conference, and also Peter highlighted that in his talk yesterday. So one example for facile processes, for ensuring operator and safety facility, and also going together with eco-friendly processes, is the job that we have done here on converting our methanol platform to a methanol-free platform, but I'm not going to talk about that anymore. We have already heard a lot of that. I want to focus a little bit more on other aspects. And if we look at on how classical Pichia strain engineering looks like, you typically start with a vector host combination screen. So first step is host strain engineering. This is not a process-specific engineering, but maybe the strain can be tailored to address some of the platform needs that you have. So the strains have improved characteristics, they are tailored to specific needs, not of a single project as said, but just covering general mechanisms to improve some of the requirements that we have. For a platform, the strains can be engineered in a way that you integrate integrative plasmids to mediate co-expression of auxiliary proteins, or you introduce some gene knockouts to improve product quality, remove host cell proteins, or improve productivity. And then you typically end up with a collection of host strains that you can screen for the aspects you desire. And as a next step, you can typically then generate the expression strains. So you're inserting the expression vectors coding for the product, for your protein of interest, and there you can vary on the codon usage of the promoters that you're testing to drive product gene expression for the secretion leader and so on. And depending on the size of the individual aspects here, you can open up a matrix of different vector host combinations, and that can be pretty labor extensive, of course, if you want to screen a lot of these combinations. So if you focus here on a particular vector host combination, and if you give it a click, then as you all know, you just click through it, please. Yeah, if you know, or as you know, then we are taking plasmids, cut them, linearize them, integrate them randomly, and we heard about the power of random integration by Tony yesterday. So you end up with a set of genetically different transformants in the end, and that makes the screening labor intensive if you want to screen a lot of different transformants in the end, because you have more or less a high degree of clonal variability between the genetically different clones. And as you all know, that clonal variability can come from copy number effects, can depend on the integration loci that are addressed, some other spontaneously occurring mutations that go together with random integration approaches, or structural variations within the chromosomes. So you basically have always two questions you need to address in a classical screening approach. So one is, what is the best vector host combination? And you need to screen for a certain number of clones in there just to get some statistical evidence on how the population performs. If you want to compare different populations, different vector host combinations, and then the aim is to select a set of good vector host combinations, and among that set, the second question is to select the best performing clones within that good performing vector host combination, but that data typically comes along already with the data you have generated at this stage. And these are then interesting clone candidates to evaluate further in a screening approach. And what I've just shown you, so identifying or generating clones, identifying interesting clone candidates is typically just the first step in a typical screening approach. Next step would be verifying the previously generated results, looking at stability, phenotypic stability, with focus on expression characteristics and quality aspects, and then final confirmation. And in a typical screening, of course, you're decreasing the number of clones you're looking at, while typically also increasing the cultivation sizes to also evaluate upscaling potential of clones, and by that also increasing the analytical efforts to properly analyze the product that's coming out of that. So I want to focus now a little bit on that aspect here, which is called phenotypic stability analysis, which is a typical working package that we are including in our clone screening approaches. And the reason for that is that we want to pretty early on already de-risk the genetic stability assessment that we need to do prior to marketing application of a new product. So that requirement, or the requirement for genetic characterization to assess genetic stability, is a requirement that comes from regulatory authorities. And what they want us to do is to have a look at the starting material of a process, which can be typically a master cell bank or working cell bank, and compare on a genetic level cells at the end of fermentation. So cells that have undergone already a lot of doublings that can sum up to 40 to 60 generations in the end when comparing starting material up to end of fermentation cell material. And what they want us to look at is a consistent copy number. If you compare starting material against end of fermentation cells, they want to ensure that there is still integrity of the cross structure of the expression construct, no insertions, deletions in there. They want to verify that the gene of interest coding sequence is still the same, and Also, assessing in general the integration sites of the expression cassettes that are put into the strain. But there are some problems or some issues or some burdensome challenges that go together with that. So if you think about random integration approaches, you typically end up, and we have also seen that in some talks before, you typically end up with expression cassettes that are serially integrated into the same locus, some concatenameric constructs, and it's not easy to assess them in multi-copy strains. You have rather long stretches of sequences that share homology. It's not easy to resolve even in long-read sequencing approaches. It's not easy to assess on the copy number. It's not easy to assess on the integration loci. Also, another aspect that goes together with verifying the gene of interest sequence is if you have multi-copy insertions, you're never sure if there are some expression cassettes that harbor some mutations, and it's not easy to assess them individually. So what people typically do is just do a PCR on genomic DNA. You have several templates of your expression cassette, you amplify that, you get a pool of PCR fragments that origin from different templates into the genomic DNA, and then just subject that to sequencing. But you don't know if there are some expression cassettes within there that harbor some mutations. You could assess that on product level with mass spec or whatever, but this is pretty labor intensive and not really feasible to do that already pretty early on in a screening approach. Then another aspect, for example, gene of interest copies that are integrated as concatenamers into the genome because of the sequence homology the different cassettes share. It's always prone that you get some loop out recombinations, so losing copy numbers when growing cells, and as I said, you can end up with 40 to 60 generations at the end of production, so there are some doublings in there where cells can lose some of the copy numbers, and then you would fail a test like that. And then another aspect is if you do serial integrations, as I said before, in a classical strain engineering approach, so you, for example, would first enter or transform cells with expression cassettes encoding for an auxiliary protein. Then in the next step, in a serial transformation event, you would enter your gene of interest expression cassette, and they always share some homology, the sequences, because all the plasmids that we are using, there are some kind of similar. You can have similar promoters, similar terminators, some other sequences within the backbone, and it's not unlikely that just as serial integration events they're interfering. So you can lose copies of your auxiliary protein expression cassette in the second transformation. These are always things that can happen and that we are also observing in real life. Okay, what we are going to do is to de-risk already that genetic stability pretty early on is, for example, looking at phenotypic stability. This is phenotypic stability with focus on production performance, and what we are doing is just mimicking GMP cell banks that you would need to do for commercial production or clinical trials in our R&D labs. So just adding additional generations within different cell banks and use them for fermentation testing and see if there's any trends, any difference in production performance. And if you do that up to a WCB level, you can already mimic like how a production process would look like in a small scale setup. And then if you do that even further in an extended cell bank, you can even look at cells that have gone beyond the limit of in vitro cell age. But nevertheless, you need to do genetic analysis of the cells. And as I said, that can be pretty labor intensive, pretty challenging if you want to do that pretty early on in a screening process. And this is actually what you wanna do, because imagine you're already spent 5, 6, 7 years for process development, and then you need to do the genetic stability assessment at the final production scale. Then it's already too late. If you go with a clone that will not pass the genetic stability assessment. And So we were thinking, how would that look like if you do targeted integration against random integration? So, and we think targeted integration would make life very much easy in terms of genetic stability analysis. I've already outlined the problems for verifying the gene of interest coding sequences in strains that were generated with random integration. If you would compare that to strains that are engineered with targeted integration, it's pretty easy to assess all the individual expression cassettes individually. You can just PCR amplify the individual cassettes because the integration loci, they are already known by design. You have sites specifically integrated them, you know where they are sitting, you can just amplify from genomic DNA, and you can have pretty easy a look at all the individual cassettes that you have integrated. You already get information from that assay for the next point. So you will get information if there are any insertions or deletions within the expression cassette, and also determination of copy number goes together with that. At least you already have an easy assay to check for the presence of the expression cassettes, if it is still there, and you can also complement the data with some pretty standard Q or digital PCR just to check if there are some additional copies integrated as a side effect. We see some further potential for targeted integration. I mean, I've outlined now how that could improve genetic stability analysis, but also going together with that is that we think that we could reduce clonal reliability because we don't have the copy number effects, we don't have effects on the integration loci, and also no effect of spontaneously arising mutations, at least that go together with the integration event. And by that, we were thinking maybe we can also streamline the screening needs. So we don't need to screen hundreds of clones to get a good one if we have a good starting point for targeted integration, but streamline the screening needs and maybe make use of that to avoid small-scale screening, but just go directly to more relevant scales that give you better predictability for future scale-up purposes. And also, this is a point which I will focus on my last slide. You can also make strain engineering steps independent from each other. So you can omit to some extent that host strain engineering concept. So you need a tool for doing targeted integration. I think we all know CRISPR is the tool of choice for doing targeted integration. We had quite some experience for E. coli engineering with CRISPR-Mat7. We have adapted that to Pichia, and we have started a small feasibility study which I want to show you. So, but first we wanted to generate a clone that we with random integration that we want to use for benchmark purposes, and we have basically undergone our typical or atypical screening process. So having done random integration, have test expressed clones in 96 well, have analyzed product formation in the supernatant by Bradford assay, which is in our hands a good proxy for product that is secreted. If you have a good performing strain, and then have done data analysis, this is how it looks like typically. So you have a distribution of clones with different production performance. Of course, you're looking at total protein at this stage. Then you need to check if the good performing ones are really good performing ones, or if these are false positives, because you could also have cell lysis that could lead to these high false positive values. And we have done so. It turned out that such clones here are false positives, but then we have identified a clone here, which was the best performing one among that set, and we used that for benchmark purposes. And as a next step, we started to work on strains with targeted integration. So we have an expression cassette for a nanobody, as shown here. We just PCR amplifying the expression cassette. with overhang primers to fuse homology regions to that. And then we have selected 11 different integration sites based on some previous knowledge, but also on some theoretical aspects that we had laid on there. And here's the data for that. So the integration loci there on different chromosomes, just to also spatially separate the integrations on different chromosomes to improve or increase genetic stability, to do not have sequences that share sequence similarity in proximity to each other. And what we have seen is that the expression performance when looking at different biological replicates is pretty similar for the different integration loci that we had identified and had tested in that approach. And we have basically selected as the next step 4 of these integration loci based on the production performance that could be deduced from that, but also on the targeting efficiency. And as a next step, we have then started to generate multi-copy strains with these integration loci. And here are the results from microtiter small-scale screening. So what you nicely see is that you have a trend of increasing product performance with increasing copy number. There we have 2 different strains that have a copy number of 2, but differ in the integration loci. You see some differences or some locus-dependent differences in the expression performance. But in general, you see that, as I said, with increasing copy number, you get more product formation. And if you benchmark that against the best clone that we got from a random integration approach, we have already seen that at least at this scale, the targeted integration strains already at pretty low copy number are equal or even superior to a clone that comes out from a random integration approach. We have then transferred clones into bioreactors, the Ember 250 system from Sartorius, and we have basically nicely seen also the same trend. So increase of productivity with increasing copy number, also the locus dependent differences for the 2 copy number strains. But interestingly, we have seen that the 4 copy number strain did not perform as good as the 3 copy number strains and did not perform as good as that strain did in the small scale expression screening. And that already shows that there is some gap in predictability when comparing small scale and bioreactor. And I mean, I've already indicated that I think this is another hint that we might even switch or skip the microtiter plate expression, but it's just feasible to do so if you can reduce your screening efforts. And targeted integration is a tool for doing so, because you can significantly lower the number of clones that you need to screen if you don't have such large effects for clonal variability. If you benchmark the random integration strain here, I think the same message as before. We are comparable or even better with the targeted integration strains, even or especially if we normalize it to biomass formation, then the 3 copy number strains is even a little superior. And we have verified the copy numbers for the targeted integration strains with digital PCR that could be confirmed, and we have also seen that that random integration strain had a copy number of 2. So I think the data fits really well. So I think we have already identified good working integration loci with that, so that 2 copy number strains from random integration and targeted integration perform the same. But I think that already shows that when you have a pool of clones that you're screening or that you put into your screening, and a good or the best performing clones only have a copy number of 2, you can just select the best clones out of that pool. But if your pool is limited, if you have no 3 copy number strains in your pool, you're never gonna be able to select a better working clone from that. And I think with targeted integration, we have much better control to systematically analyze effects like that may come from copy number. Okay, then the last slide to give you some insight on the freedom you have in the order of modifications for introducing into strains for strain engineering. Typically you have, or you want to introduce your product gene, and you want to have a toolbox of genetic modules that you can plug into your strain for strain engineering. That might be overexpression of auxiliary proteins, gene knockouts, or some other modules that may help to improve processability. And if you give it a click, with random integration and with the platform strain approach, if you want to have consistency between strains of, for example, individual projects, you always need to follow the certain order of modification events, or you always need to rely on that particular platform strain that you have generated before with random integration. Otherwise, you're never going to be able to reproduce that. You're never going to have the same copy number of your auxiliary proteins at the same spot sitting in the chromosome. So it's very hard to have some consistency between individually generated strains. But I think that goes with targeted integration, because you can just plug in the different modules at the site and at the time you want to do it within your strain engineering approach. So I've just outlined a random approach here. So for example, you start with a wild type cell, you first integrate in a module an auxiliary protein of that you know that will always help to express your protein of interest for a particular molecular class. You could use that as a kind of platform. Then you're just, as the next step, you could assess copy number effects. You might see that a 3 copy number strain is the best working one because it is better than the 2 copy strain and the 1 copy number strain. And then you can just decide on what is the next step. You could then, as a next step, take your 3 copy number strain, include a 4th copy, assess if that one is even better, or you can just take some other strains that you have generated before, plug in some additional functional modules to check if there's some improvement in quality aspects you have seen, or to circumvent some quality issues that have become obvious in a formal screening. As a next step, if you have that assessed individually, you can combine your gain of knowledge. You can have a 4 copy number strain with additional modules. And in that approach, it doesn't matter when you're doing the different modifications. And I think that is pretty nice. And I think also another side effect is that the strains coming out of that are all antibiotic or even oxotrophy marker free. So that can be also helpful for some certain applications. With that, I'm on the end, and I want to thank all the people at Sanofi working on our Pichia platform, working on improving our Pichia platform at both sites we're dealing with nanobody, the colleagues from Ghent, and also the colleagues from Frankfurt. Thank you.
Questions? Nice presentation, really, really interesting for me. So when you are using Pichia pastory, and you want to do CRISPR-Cas, there's a big problem that you probably know. The problem is that Pichia pastory depend more on non-homologous end joinings. There's a big problem. If you compare, for example, with Saccharomyces is really very nice to do CRISPR-Cas. How did you solve this problem when you use CRISPR-Cas?
I think, to our experience, CRISPR-Cas is already the key for ensuring that. I think the problem that you're describing is more related if you want to do just the non-CRISPR assisted integration. I think that often goes together with some off-target effects, with some off-target non-site specific integrations. But in our hands and to our experience, CRISPR already helps very much in having site-specific integrations.
Can you comment on when you do single chromosomal isolation, you're process?
That is basically done at the end of the screening approach. So then we are doing the documented clonality, or we are doing the singling out and integrating the documented clonality step. I mean, isolation is already done after transformation, but that is not really documented then, and it's not really ensured. But then we are doing it after we have identified at least a small set of interesting clone candidates. So that would be at the end of the screening approach or screening workflow that I've shown.
Can you comment how many promising clones you need to dump because of lack of genetic stability? Roughly?
Yeah. I mean, it's hard to say. It really also depends on how the expression construct looks like, but I can say that it's not a rare event. It happens often quite some time. This is why we also want to tackle that issue with these approaches. Yeah.
One last short.
In one of the previous talks, there was talk about jackpot clones. I know obviously your approach works well to get the consistency within a box, but can you comment on maybe getting something outside of the box? Are you interested in that?
Yeah, perfect question. And I think both approaches, so identifying jackpot clones with random integration and targeted integration, these is no contradictory approaches, but I think they go together. I've said that, for example, for a platform strain, you can also introduce some gene knockouts. These can be, for example, gene knockouts that have been identified previously in a random integration approach and were deduced from jackpot clones. And I think that is also the basis that we need to create for targeted integration. Because, I mean, you have seen, and I've also said, we are aiming to reduce screening efforts because we have a better homogeneity between the different clones that we are generating with targeted integration. But of course, we don't want to be the clones homogeneously poor expressing. We want to have them homogeneously well expressing, and therefore you need to find the right starting point, the right basis to build on your strain engineering. And I think all the findings from random integration from jackpot clones, that feeds in there. Yeah.